Hello. In today's video, we shall learn in detail about lichens. By the end of this video, you will have a clear understanding about its types, internal structure, specialized structures, asexual and sexual modes of reproduction, and also about the fruiting body, apothecium. Let us start with the introduction to lichens. The branch of botany that deals with the study of lichens is called lichenology. The thallus of every lichen is composed of two different organisms, a fungus or the mycobiont and an alga or phycobiont living in symbiotic association. The algal partner is mostly a green alga or a cyanobacterium. The fungus and the alga in the lichen thallus derive mutual benefit from their partnership. The fungus with the rhizoids absorbs water and minerals from the substratum which are passed on to the alga. In addition, the fungal hyphae provide shelter to the alga. The alga in return synthesize carbohydrates with the help of chloroplast and supplies food to the fungus. There are three main types of lichen thalli on the basis of their morphological structure. They are crustose lichens, foliose lichens, and fruticose lichens. Crustose lichens. The thallus is flat, thin, with no distinct lobes. It is closely attached by the lower surface to stones or rocks. The surface of the thallus shows hexagonal areas called the areolae, example Graphis crypta. Foliose lichens, the thallus is flat, broad, lobed and leaf-like. It has a distinct upper and lower surface. The foliose lichen thallus is attached to the substratum by rhizoid-like structures called rhizinae, which are present on its lower surface. The free end of the rhizine forms a disc which secretes mucilage and acts as an anchorage and absorptive organ. Example, Xanthoria. Fruticose lichens, the thallus has a complex structure. It is slender with cylindrical or ribbon-like branches which may be erect or pendant and tassel-like. The thallus is attached only at the base by a flattened disc. Example, Usnea. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us continue with lichen thallus. Depending on the growing region, lichens are grouped into corticoles, saxicoles, and terricoles. Corticoles grow on bark of trees and are mostly confined to the tropical and subtropical region where there is abundance of moisture. Saxicoles grow on rocks in cold regions. The terricoles are terrestrial and inhabit the soil. Based on the kind of fructification of the fungal partner, the lichens are divided into different groups, namely ascolichens and basidiolichens. In ascolichens, the fungal component is an ascomycete. It is further subdivided into gymnocarpiae, in which the ascocarp is of an apothecium type, and pyrenocarpiae, in which the ascocarp is perithecium type. In basidiolichens, the fungal component is a basidiomycete. Let us look into the internal structure of lichen thallus. Based on the distribution of algal member inside the thallus, the lichens are divided into two groups, namely homoisomerous and heteromerous lichens. Structure of homoisomerous lichen thallus. The thallus of Collima species shows a simple structure with hardly any differentiation. The fungal hyphae is loosely interwoven and the algal cells are equally distributed and scattered between the fungal hyphae. Structure of heteromerous lichen thallus. The thallus of Xanthoria shows distinct zones with algal component restricted to a specific zone. The four zones are upper cortex, algal zone, medulla, and lower cortex. The upper cortex, it is thick and protective, with fungal hyphae forming a compact, interwoven, tissue-like layer called pseudoparenchyma. Intercellular spaces may or may not be present. Algal zone. This zone lies beneath the upper cortex. The algal cells are intermixed with loosely interwoven fungal hyphae. Medulla. It is the central core of the thallus. The fungal hyphae are scattered and loosely interwoven. They run in all directions. Lower cortex. It is the lower surface of the thallus in which the fungal hyphae are densely packed. Rhizinae arise from this layer and penetrate the substratum to serve as anchoring organ. Next, let us learn about the specialized structures of lichen thallus. Breathing pores. In certain lichens, the fungal hyphae in the upper cortex become loosely interwoven at certain places, forming breathing pores which help in aeration. Cyphalae. In certain lichens, there are cup-like white spots in the lower cortex. These spots are cavities or concave circular depressions consisting of white medulla. Here, the hyphae grow directly from the medulla and form empty, rounded cells at their tips. When these aerating or breathing pores have a definite border, they are called cyphalae. When border is absent, these breathing pores are known as pseudocyphalae. Cephalodia. In certain lichens, there are hard, gall-like swellings on the free surface. The cephalopodium contains the same fungal hyphae as in the thallus, but the algal component is different. Isidia, on the upper surface of the lichen thallus, there are small outgrowths which consists of an outer cortical layer and an inner algal layer. The algal component is similar to the one present in the thallus. They help to increase the surface area for photosynthesis. The isidia may be rod-shaped, cigar-shaped, coral-like buds, or scale-shaped. Next, we shall study about the reproduction seen in lichen thallus. Lichens reproduce both asexually and sexually. Asexual reproduction may take place by vegetative methods or by sporulation. The algal cells reproduce by simple fission and the fungus produces spores. Vegetative methods of asexual reproduction. First, fragmentation. It is an efficient means of propagation. The thalli may break into several segments either due to aging or due to certain accidents. The segments that contains both the symbionts develop into a new lichen. Thus, a number of separate individual lichen, all derived from the parent thallus, are formed. Next is Soredia. These are reproductive bodies containing both the algal component and the fungal hyphae of the lichen thallus. They are small, rounded granules or bud-like outgrowths seen on the upper surface of the thalli. Each soredium contains few algal cells closely surrounded by fungal hyphae similar to the parent thallus. Sometimes soredia may occur as pustule-like well-organized structures, then it is known as soralia. Soredia are produced from the algal component of the lichen thallus. The soredia are exposed when the fungal hyphae elongates, resulting in the rupture of the cortex. They are easily dispersed by wind. They may form a grey coating of soredial dust on the trees. On reaching a suitable substratum, each soredium germinates and grows into a new lichen thallus. Next, isidia. Certain lichens show the presence of small conical warts on the thallus. It has an outer cortical layer followed by an algal layer. It can be easily cut off from the parent thallus and during favorable conditions grows into a new lichen thallus. Sporulation. The fungal component produces small, non-motile, asexual spores called pycnidiospores in conical flask-shaped cavities called pycnidia. Each pycnidium has a small pore or opening called the ostule. The pycnidium wall contains sterile fungal hyphae. From the wall arises fertile hyphae which produces asexual spores, pycnidiospores, at their tips. These spores on germination produce the fungal hyphae which form new lichens if they come in contact with their algal partner. Next, we shall study about sexual reproduction in the fungal component. The male reproductive organ of the fungal partner is the spermogonium and the female reproductive organ is the carpogonium. Spermogonium. Each spermogonium is a flower-shaped receptacle formed on the upper surface of the thallus with a small pore and ostiole at the surface. The spermogonium has both fertile and sterile hyphae. The fertile hyphae at the tips form male non-motile cells called the spermatia. Each spermatium is surrounded by a cell wall and is released through the ostiole. Carpogonia. The carpogonium consists of a lower coiled portion called the ascogonium and an upper straight portion called the trichogyne. The ascogonium is multicellular, uninucleate, lies in the medullary region of the thallus. The trichogyne is multicellular with elongated cells. The terminal cell of the trichogyne projects above the surface of the thallus. Next is fertilization. The spermatia or the male cells adhere to the gelatinous wall of the terminal cell of the trichogyne. The intervening walls between the spermatium and the trichogyne dissolve and the contents of the spermatium move into the trichogyne. Post-plasmogamy changes. After fertilization, various changes are seen in the carpogonium. The trichogyne withers and branched ascogenous hyphae develop from the ascogonium. Asci are formed at the end of the ascogenous hyphae. The ascus fruit is either of apothecium type or of perithecium type. Next is the structure of apothecium. The apothecium is rounded, cup-shaped fruit body of an Ascomycetes fungus. The apothecium consists of two parts, namely the disc and the margin. Disc of the apothecium. It is the fertile portion of the apothecium. It consists of club-shaped asci between the sterile fungal hyphae paraphysis. The asci and the paraphysis form a continuous, closely packed, fertile layer called the hymenial layer or hymenium or thecium. Beneath the hymenium is a dense mass of sterile hyphae called the subhymenium, from which arises the paraphysis. The projecting ends of the paraphysis forms a smooth surface known as the epithecium. Margin of the apothecium surrounding the disc and forming the edge of the apothecium is the proper margin. Development of ascospores. The diploid nucleus in the ascus mother cell, formed by the fusion of two haploid nuclei, undergoes meiosis and mitosis, resulting in 8 haploid nuclei. These nuclei are surrounded by cytoplasm and form the ascospores. As the ascus swells by absorbing water, it bursts and the ascospores are liberated and carried by wind to distant places. On reaching the suitable soil, the ascospores germinate and produce fungal hyphae. When the fungus comes in contact with its algal partner, it forms a new lichen thallus. In Pyrenocarpae, the ascus fruit is of perithecium type. The perithecium is a flower-shaped structure and its cavity is lined with asci. The paraphyses, if present, are very few in number. So today, we have learnt in detail about different types of lichens, their internal structure, as well as certain specialized structures, their various modes of reproduction, and structure and development of fruiting body apothecium. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.